Welcome. Welcome to my channel, Deep Learning Biology. My name is Dr. Vikash Bhardwaj. Now in this video, I am going to teach you how enzymes they work. So in this video, you are going to learn how enzymes they actually work. Try to see here. You must be knowing enzymes, they are known as biocatalyst. And generally what they do, they enhance the rate of reactions. Try to see here. Let's suppose you are carrying out one reaction in absence of enzyme. Okay, let's suppose there is two substrate A plus B. They are interacting, and let's suppose you are carrying out the reaction in absence of enzyme. Then it may take millions of years to complete that reaction. In certain situation, in absence of enzymes, it may take millions of years to get the desired product. But in presence of enzyme, okay, the same reaction can be completed very fast. Okay, here hopefully you know enzymes, they are biocatalysts, and what generally they do, they enhance, they accelerate, okay, they enhance the rate of reactions. And generally they do this very specifically. For one particular enzyme, you are going to have one particular substrate. So here enzymes, what generally they do, they convert substrate into product. But here I also want to tell you, please keep in mind that enzymes, they also convert the product into substrate. So here keep in mind that enzymes, they actually carry out the interconversion of substrate into product into substrate. So here try to see, yes, enzymes, they are known as biocatalysts. They enhance the rate of reaction, they accelerate the rate of reaction. The most important point, keep in mind, the enzymes, they carry out interconversion of substrate to product. Yes, they can also carry out conversion of product to substrate. This is very much important. Now let us see how enzymes, they actually work. Try to see here. Okay, if you see any enzyme, okay, let's suppose we are going to have enzyme like this. Let's suppose this is your substrate. This is your substrate. Now here initially what is going to happen, the substrate will bind to the enzyme, and within the enzyme you are going to have a pocket, and this is known as active site. So here initially what is happening, the substrate, okay, it is going to bind to the active site. Okay, now here once the substrate is going to bind to the active site, yes, there is conformational change within the enzyme, and that is known as induced fit. There is change in the shape of the enzyme, there is change in the pocket of the enzyme. Now here actually try to see what is happening. Okay, yes, at the last once the reaction is complete, what you are going to see, the release of product. Okay, but try to see what is happening when the substrate is going to bind to the enzyme active site. Here, try to see, initially your substrate, okay, it is being surrounded by a layer of water. Try to see within the cell, okay, you see within the cell, if you see that there is 70 to 75% water. So around the substrate also, what you are going to see, a layer of water. Now here you are going to have your enzyme, okay, there is a pocket, okay. Initially what is going to happen, these substrate, okay, they will bind at the pocket, okay. So here what is going to happen initially, okay, there will be a removal, you can say the desolvation of water layer. So first thing is that, yes, when the substrate is going to bind at the active site, first thing is that there is going to be desolvation of water molecules. So there is removal of water molecules which are around the substrate. So here, what will be the effect? Try to see here. Here you see enzyme and the substrate. Enzymes, they are comparatively larger in size, and here your substrate, it is very small in size. Okay. Yes, here if you take a buffer, okay, let's suppose within the buffer you are going to have the substrate. Okay, now these substrate, okay, let's suppose here you are having the substrate glucose, you are also having another substrate, okay, ATP, okay. You have two things, glucose and ATP. No reaction is going to happen, no reaction. But if you add the enzyme, specific enzyme, okay, like hexokinase, okay, the reaction will happen and there will be synthesis of glucose 6-phosphate. Just if you are going to have glucose and ATP, no reaction will happen. If you add hexokinase in presence of suitable buffer, what is going to happen? Okay, the glucose, it will get converted into glucose 6-phosphate. Now try to see here, initially glucose and ATP molecules. Okay, so around the glucose, okay, around the ATP, you are going to have water layer. Okay, another important thing, all these molecules of glucose and ATP, they are moving randomly. They are not coming in contact of each other in a proper way. So here, when you are going to add one enzyme, so within the pocket, okay, the substrate will bind, and around the substrate, what you are going to have? You are going to have layer of water, and this layer of water will be removed. So here, at the first step, what is going to happen? There is desolvation of water which is present around the substrate. Okay. Now here try to see, when your substrate, it is in this pocket, try to think the situation when your substrate was moving randomly. Here you see in this situation, it was moving randomly. Here the entropy is high. Entropy is degree of randomness. In this situation when no enzyme is being added, what is there? The entropy is high. But once your substrate, it is within the pocket, okay, so what is going to happen? What you are going to see, the entropy, it is going to reduce. It is just confined to this area. Now it can't move rapidly. Here it can go in this direction, this. Okay, so here in this situation entropy is high, and here when the substrate is going to bind within the pocket, the entropy, it is going to reduce. Yes, within the enzyme, okay, here you see within the pocket of the enzyme you are going to have amino acid, and generally these amino acids, they are going to interact with the substrate. Okay, the amino acids, they are going to interact with the substrate. Okay, so here the amino acids, they can interact. Most of the times the interactions are non-covalent interaction. Non-covalent interaction, like you are going to have hydrophobic interaction, like you are going to have ionic interaction, okay, and you are going to have hydrogen bonds, okay, etc., van der Waals interactions. Okay, so initially what is going to happen? In between substrate and the enzyme, there is going to be non-covalent interaction. Yes, there can be also covalent interactions also. Okay, in covalent interaction, you are going to see formation of covalent bond. But here keep in mind, generally the covalent interaction, they are going to decide path of the reaction. They are going to decide how the substrate will be converted into product, by which chemical way. But see here, these non-covalent interaction, okay, these are actually the weak forces. Now what they will try, okay, these weak forces, they will try to make enzyme substrate complex. So here initially enzyme will go and bind to the substrate will go and it bind to the enzyme. What is going to happen? There is formation of enzyme substrate complex. Here keep in mind these non-covalent interaction, the weak forces, okay, here during the time of formation of enzyme substrate complex, there will be break and formation of bonds. Okay, and here I have told you, once the enzyme, once the substrate is going to bind to the pocket, initially what is going to happen? There will be decrease in the entropy. There will be decrease in the randomness. Yes, here this break and formation of bonds, the weak bonds, okay, it is going to release energy. That energy is known as binding energy. Okay, so here what is going to happen? That due to non-covalent interaction, okay, what is going to happen? There will be breakage and formation of bonds, and all these situations, they are going to release binding energy. This binding energy, it is going to lower down activation energy. It is going to lower activation energy. Try to see here. This is very much important. Here try to see. Let's suppose you are going to have one, okay, substrate. So substrate is going to have some free energy, Okay, so here we are going to denote the free energy. Okay, so substrate is going to have some free energy. Now let's suppose this substrate, it is being converted into product. Okay, so here this is the free energy of the product. Now try to see here. Here your substrate is going to have much higher free energy. Your product is going to have lower free energy. Okay, so here if you see change in Gibbs free energy, it will be negative. So this is reaction coordinates. If you see the change in Gibbs free energy, this delta G, it is negative. The reaction must occur on its own. No. Try to see here. To convert substrate into product, there is a barrier. There is a barrier. The substrate has to initially convert into a transition state, then it has to convert into a product. Okay, now here this is the barrier. It has to reach one transition state. To reach the transition state, the substrate need, okay, this amount of activation energy. This is EA, activation energy. How the reaction is going to produce? How the reaction is going to proceed? The substrate, okay, it has to reach a transition state. Remember, this transition state is not a stable state. And to reach this state, okay, it needs activation energy. It needs activation energy. Now, here I just want to tell you, enzymes, okay, when there is a presence of enzyme, okay, when this substrate is going to bind to the pocket within the enzyme, what will happen? It will lower down the activation energy. So when the binding energy is released, that energy will be used to lower down the activation energy. So here, remember, keep in mind, enzymes, they work, enzymes, yes, they work specifically, they work at a very fast rate. How? Because enzymes, they can lower down the activation energy. Remember, in absence of enzymes, sometimes a reaction may take millions of years, but in presence of enzyme, the same reaction can happen at physiological pH, at physiological temperature, okay, because the enzymes, they can lower down the activation energy. And how the enzymes are going to lower down the activation energy? Because there is a release of binding energy. And this binding energy actually lower down the activation energy. So hopefully through this video, you have learned how enzymes, they actually work. Please do like, share the video and subscribe to my channel, Deep Learning Biology. Bye bye. Take care.